Hello everyone, here is the first class of genetic engineering by Dr. Ali Mouahedi, presented at Nanjing Pharsi University. Okay, here we go. Firstly, we want to explain about chapter 1, what is genetic engineering. In this chapter, we have some summaries. You can see here what is genetic engineering, laying the foundations, first steps, and finally concept map. And also we will define genetic engineering as it will be described to outline the basis features of genetic engineering, and we will describe the emergence of gene manipulation technology. So what is genetic engineering? There are several other terms that can be used to describe the technology, including gene manipulation, gene cloning, recombinant DNA technology, genetic modification, and the new genetics. Although there are many diverse and complex techniques involved, the basic principles of genetic manipulation are reasonably simple. The premise on which the technology is based is that genetic information encoded by DNA and arranged in the form of genes is a resource that can be manipulated in various ways to achieve certain goals in both pure and applied science and medicine. The principle of genetic manipulation is the ability to isolate a single DNA sequence from the genome. This is the essence of gene cloning and can be considered as a series of four steps. You can see the four steps of genetic engineering in this figure 1.1. So successful completion of these steps provides the genetic engineer with a specific DNA sequence, which may then be used for a variety of purposes. A useful analogy is to consider gene cloning as a form of molecular agriculture, enabling the production of large amounts in genetic engineering. This means micrograms or milligrams of a particular DNA sequence. Even in the era of large-scale sequencing projects, this ability to isolate a particular gene sequence is still a major aspect of gene manipulation carried out on a day-to-day basis in research laboratories worldwide. So here is laying of foundations. Although the techniques used in gene manipulation are relatively new, it should be remembered that development of these techniques was dependent on the knowledge and expertise provided by microbial genetics. We can consider the development of genetics as falling into three main eras. Figure 1.2 is describing three eras for genetic engineering. This is the history of genetics science for genetic engineering scientists. So the science of genetics really began with the discovery of George Mendel's work at the turn of the century, and the next 40 years or so saw the elucidation of the principles of inheritance and genetic mapping. Microbial genetics became established in the mid 1940s, and the role of DNA as the genetic material was confirmed. During this period, Great advances were made in understanding the mechanisms of gene transfer between bacteria, and a broad knowledge base was established from which later developments would emerge. The discovery of the structure of DNA by James Watson and Francis Crick in 1953 provided the stimulus for development of genetics at the molecular level, and the next few years saw a period of intense activity and excitement as the main features of the gene and its expression were determined. So, the first steps in the late 1960s, there was a sense of frustration among scientists working in the field of molecular biology. Research had developed to the point where progress was being hampered by technical constraints, as the elegant experiments that had hoped to decipher the genetic code couldn't be couldn't be extended to investigate the gene in more detail. However, a number of developments provided the necessary stimulus for gene manipulation to become a reality. In 1967, the enzyme DNA ligase was isolated. This enzyme can join two strands of DNA together, a condition for the construction of recombinant molecules, and can be regarded as a sort of molecular glue. This was followed by the isolation of the first restriction enzyme in 1970, a major milestone in the development of genetic engineering. Restriction enzymes are essentially molecular scissors that code DNA at precisely defined sequences. Such as enzymes can be used to produce fragments of DNA that are suitable for joining to other fragments. Thus, by 1970, the basic tools required for the construction of recombinant DNA were available. The first recombinant DNA molecules were generated at Stanford University in 1972, utilizing the cleavage properties of restriction enzymes and the ability of DNA ligase to join DNA strands together. The importance of these first tentative experiments cannot be overestimated. Scientists could now join different DNA molecules together and could link the DNA of one organism to that of a completely different organism. The methodology was extended in 1973 by joining DNA fragments to the plasmid PSC101, which is an extra chromosomal element isolated from the bacterium Escherichia coli. These recombinant molecules behaved as replicants, that is, they could replicate when introduced into E. coli cells. Thus, by creating recombinant molecules in vitro and placing the construct in a bacterial cell where it could replicate in vivo, specific fragments of DNA could be isolated from bacterial colonies that formed clones. Colonies are formed from a single cell, in which all cells are identical. When grown on agar plates, this development marked the emergence of the technology that became known as gene cloning. So here in figure 1.3, you can see the cloning steps. Cloning DNA fragments. A, you can see the source DNA that will be isolated by enzymes into fragments into suitably sized pieces. Then the fragments are joined to a carrier molecule or vector to produce recombinant DNA molecules. In this case, a plasmid vector is shown. So the recombinant DNA molecules are then introduced into its host cell, for example, a bacterial cell in this example, for propagation as clones. Okay, here is the concept map. Totally, genetic engineering is an enabling technology that involves cutting, modifying, and joining DNA molecules using what? Using enzymes. For example, DNA ligase or restriction enzymes. So, using restriction enzymes can be used for generation of DNA fragments. So after that, the generated DNA fragments will be joined together or to vector or carrier molecules using DNA ligase. So these vectors or recombinant DNA introduced into a host cell for selection of desired sequence. Also, the genetic engineering is known as gene cloning, recombinant DNA technology, molecular cloning, and etc. So, that has application in pure science, biotechnology, and medicine for legal and ethical questions. So arose from microbial and molecular genetics and was first achieved in 1972 at the Stanford University. Okay, let's go to the chapter 2, Introducing Molecular Biology. In this chapter, and as a summary, we have to illustrate how living systems are organized at the genetic level. To describe the structure and function of DNA and RNA, and also we have to outline the organization of genes and genomes. Also, we will describe the processes involving gene expression and its regulation. The way that living systems are organized. Before we look at the molecular biology of the cell, it may be useful to think a little about what cells are and how living cells systems are organized. Two premises are useful here. First, there is a very close link between structure and function in biological systems. And second, living systems provide an excellent example of the concept of emergent properties. The chemistry of living systems is based on the element carbon, which can form four covalent bonds with other atoms. By joining carbon atoms together and incorporating other atoms, molecules can be built up, which in turn can be joined together to produce macromolecules. Biologists usually recognize four groups of macromolecules. First, lipids. Second, carbohydrates. Third, proteins. And finally, nucleic acids. The synthesis of macromolecules involves a condensation reaction between functional groups on the molecules to be joined together. This dehydration synthesizes forms a covalent bond by removing the elements of water. In the case of the large polymeric macromolecules of the cell, polysaccharides, proteins, and nucleic acids, hundreds of thousands or even millions of individual non-numeric units. Okay, in the case of the large polymeric macromolecules of the cell, polysaccharides, proteins, and nucleic acids, for hundreds of thousands or even millions of individual monomeric units may be joined together in this way. The polymers can be broken apart into their constituent monomers by adding the elements of water back to reconstitute the original groups. This is known as hydrolysis. Literally, hydro means water and lysis means breaking. The monomer-polymer cycle and dehydration hydrolysis are illustrated in Figure 2.1. So here for example, for monomers, we have amino acids that we releasing one molecule of water, so or dehydration, so it could be condensed and make a polymer by by 100,000 or sometimes millions of amino acids. Anyway, this protein or polymer will attract again water or hydrolysis and will be again like as monomer. So you can see here the dehydration hydrolysis can make a cycle inside a cell. So the monomer polymer cycle in this example, a representation of amino acids and proteins is shown. So the amino acids or monomers are joined together by removal of the elements of water during dehydration synthesis. When the protein is no longer required, it may be degraded by adding back the H2O during hydrolysis. Although the cycle looks simple when presenting like this, the synthesis of proteins requires many components whose functions are coordinated during the complex process of translation. So the cell is the basic unit of organization in biological systems. Although there are many different types of cell, there are some features that are present in all cells. There is a cell membrane, the plasma membrane, that is the interface between the cell contents and the external environment. Some cells, such as bacteria, yeasts, and plant cells, may also have a cell wall that provides additional structural support. Some sort of genetic material, or almost always DNA, is required to provide the information for cells to function, and the organization of this genetic information provides one way to classifying cells. In prokaryotic cells, for example, bacteria, the DNA is not compartmentalized. Whereas in eukaryotic cells, the DNA is located within a membrane-bound nucleus. So the eukaryotic cell also utilize membranes to provide additional internal structure. Prokaryotic cells are generally smaller in size than eukaryotic cells, but all cells have a maximum upper size limit. This is largely because of the limitations of diffusion as mechanisms for gas and nutrient exchange. Typically, bacterial cells have a diameter of 1 to 10 micrometers, and plant and animal cells are 10 to 100 micrometers. The expression of genetic information or the flow of genetic information to set the structure of nucleic acids in context. It is useful to think a little about what is required in terms of genetic information enable a cell to carry out its various activities. It is a remarkable fact that an organism's characteristics are encoded by a four-letter alphabet defining a language of three-letter words. The letters of this alphabet are the nitrogenous bases adenine or A, guanine or G, cytosine or C, and thymine or T. So, how do these bases enable cells to function? The expression of genetic information is achieved ultimately via proteins, particularly the enzymes that catalyze the reactions of metabolism. Proteins are condensation heteropolymers synthesized from amino acids, of which 20 are used in natural proteins. As a basis or critical informatic components, we can calculate the using the basis singly would not provide enough scope. Only four possible arrangements to encode 20 amino acids, as there are only four possible code combinations A, G, C, and T. If the bases were arranged in pairs, that would give us a 4 power 2 or 16 possible combinations are still not enough. Triplet combinations provide 4 power 3 or 64 possible permutations, which is more than sufficient. Thus, great diversity of protein form and function can be achieved using an elegantly simple coding system with set of three nucleotides or codons specifying the amino acids. Thus, a protein of 300 amino acids would have a minimum coding requirement of 900 nucleotides on a strand of DNA. The genetic code or dictionary is one part of molecular biology that, like the double helix has become something of a biological icon. Although there are more possible codons that are required, or 64 as opposed to 20. Three of these are stop codons. Several amino acids are specified by more than one codon, which accounts for the remainder, a feature that is known a redundancy of the code. So codons read 5' to 3', thus A, U, and G means methionine as a start codon. So the three-letter abbreviations for the amino acids are as follow: ALA, alanine; ARG, arginine; ASN, asparagine; ASP, aspartic acid; CYS, cysteine; GLN, glutamine; GLU, glutamic acid; GLY, glycine; HIS, histidine; ILE, isoleucine; LEU, leucine; LYS, lysine; MET, methionine, PHE, phenylalanine, PRO, proline, SER, serine, THR, threonine, TRP, tryptophan, TYR, tyrosine, VAL, valine. So the three codons UAA, UAG, and UGA specify no amino acid and terminate translation. The flow of genetic information is unidirectional from DNA to protein with messenger RNA or mRNA as an intermediate. The copying of DNA-encoded genetic information into RNA is known as transcription, with the further conversion into protein being termed translation. So, this concept of information flow is known as the central dogma of molecular biology and is an underlying theme in all studies of gene expression. To further aspects, the information flow may be added to this basic model to complete the picture. First, duplication of the genetic material prior to cell division represents a DNA to DNA transfer known as DNA replication. A second addition with important consequences for the genetic engineer stems from the fact that some viruses have RNA instead of DNA as their genetic material. These viruses, chiefly members of the retroviruses group, have an enzyme called reverse transcriptase. And RNA, so the reverse transcriptase is an RNA-dependent DNA polymerase that produces a double-stranded DNA molecule from the single-stranded RNA genome. Thus, in these cases, the flow of genetic information is reversed with respect to the normal convention. The central dogma is summarized in Figure 2.2. You can see here from DNA to mRNA to protein, the processes of transcription or TC, translation TL, and DNA replication or I obey this rule. An exception is found in retroviruses or RNA viruses, which have an RNA genome and carry out the process known as reverse transcription or RTC to produce a DNA copy of the genome following infection of the host cell. In most organisms, the primary genetic material is double-stranded DNA. What is required of this molecule? First, it has to be stable, and as genetic information may need to function in a living organism for up to 100 years or more. Second, the molecule must be capable of replication to permit dissemination of genetic information as new cells are formed during growth and development. Third, there should be the potential for limited alternation to the genetic material or mutation to enable evolutionary processes to exert their effects. The DNA molecule fulfills these criteria of stability, replicability, and mutability, and when considered with RNA provides an excellent example of the premises. that we considered earlier. The very close relationship between structure and function, and the concept of emergent properties. Nucleic acids are heteropolymers composed of monomers known as nucleotides. A nucleic acid chain is therefore often called a polynucleotide. The monomers are themselves made up of three components: a sugar, a phosphate group, a sugar, a phosphate group, and a nitrogenous bases. So the two nucleic acids are polymers types of nucleic acid, DNA and RNA, are named according to the sugar composed of nucleotides, with DNA having 2' deoxyribose as the deoxyribonucleic acid. RNA RNA is ribonucleic acid having ribose. So, the sugar phosphate components of a nucleotide are important in determining the structural characteristics of polynucleotides, and the nitrogenous bases determine the nitrogenous bases determine their information storage and transmission characteristics. So here, the structure of a nucleotide is summarized. The structure of a nucleotide as carbon atoms are represented by solid circles. Carbon atoms numbered 1 to 5. In DNA, the sugar is deoxyribose with a hydrogen atom at position X. In RNA, the sugar is ribose, which has a hydroxyl group at position X. The base can be A, G, C, or T in DNA and A, G, C, or U in RNA. Nucleotides can be joined together by a 5' to 3' phosphodiester linkage, which confers directionally on the polynucleotide. Thus, the 5' end of the molecule will have a free phosphate group, and the 3' end a free hydroxyl group. This has important consequences for the structure, function, and manipulation of nucleic acids. In a double-stranded molecule such as DNA, the sugar phosphate chains are found in an antiparallel arrangement, with the two strands running in different directions. So, the nitrogenous bases are the important components of nucleic acids in terms of their coding function. In RNA, the base thymine is replaced by uracil, which is functionally equivalent. Chemically, adenine and guanine are purines, which have a double ring structure, whereas cytosine and thymine or uracil are pyrimidines, which have a single ring structure. In DNA, the bases are paired. The bases are paired A with T and G with C. This pairing is determined both by the bonding arrangements of the atoms in the bases and by the special constraints of the DNA molecule. The only satisfactory arrangement being a purine pyrimidine base pair. The bases are held together by hydrogen bonds, two in the case of an A to T base pair and three in the case of a G to C base pair. So, the structure and base pairing arrangement of the four DNA bases is shown here in 2.4. The DNA molecule in vivo usually exists as a right-handed double helix called a B form. This is the structure proposed by Watson and Crick in 1953. Alternative forms of DNA include the A form, or right-handed helix, and the Z form, left-handed helix. Although DNA structure is a complex topic, particularly when the higher order arrangements of DNA are considered, a simple presentation will suffice here, as shown in Figure 2.5. The double helix DNA, this is DNA in the commonly found B form. The right-handed helix has a diameter of 2 nanometers and a pitch of 3.4 nanometers with 10 base pairs per turn. The sugar phosphate backbones are antiparallel, so you can see here the arrow backbones are antiparallel arrow with respect to their 5' to 3' orientations. One of the sugar phosphate chains has been shaded for clarity. So the pyrimidine base pairs are formed across the axis of the helix. So the structure of RNA is similar to that of DNA. The main chemical differences are the presence of ribose instead of 2' deoxyribose and uracil instead of thymine. RNA is also most commonly single-stranded, although short stretches of double-stranded RNA may be touched in self-complementary regions. There are three main types of RNA molecule found in cells: messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. Ribosomal RNA is the most abundant class of RNA molecule, making up some 85% of total cellular RNA. It is associated with ribosomes, which are an essential part of the translational machinery. Transfer RNAs make up about 10% of total RNA and provide the essential specificity that enables the insertion of the correct amino acid into the protein that is being synthesized. Messenger RNA, as the name suggested, acts as the carrier of genetic information from the DNA to translational machinery and usually makes up less than 5% of total cellular RNA. The gene can be considered the basic unit of genetic information. Genes have been studied since the turn of the century when genetics became established. Before the advent of molecular biology and the realization that genes were made of DNA, the study of the gene was largely indirect. The effects of genes were observed in phenotypes and the behavior of genes was analyzed. Despite the apparent limitations of this approach, a vast amount of information about how genes functioned was obtained, and the basic tenets of transmission genetics were formulated. So, For gene organization, the term gene is usually taken to represent the genetic information transcribed into a single RNA molecule, which is in turn translated into a single protein. Exceptions are genes for exceptions are genes for RNA molecules such as rRNA and tRNA, which are not translated. In addition, the nomenclature used for prokaryotic cells is slightly different because of the way that their genes are organized. Genes are located on the chromosomes, and the region of the chromosome where a particular gene is found is called the locus of that gene. In diploid organisms, which have their chromosomes arranged as homologous pairs, different forms of the same gene are known as alleles. The double-stranded DNA molecule has the potential to store genetic information in either strand, although in most organisms, only one strand is used to encode any particular gene. So there is the potential for confusion with the nomenclature of the two DNA strands, which may be called coding or non-coding, sense or antisense, plus or minus, transcribed or non-transcribed, and finally template or non-template. Recommendations from the International Union of Biotechnology and International Union of Pure and Applied Chemistry favor the terms coding or non-coding, with the coding strand of DNA taken to be the mRNA-like strand. The terms template and non-template will be used to describe DNA strands when there is not necessarily any coding function involved, as in the copying of DNA strands during cloning procedures. Thus, genetic information is expressed by transcription of the non-coding strand of DNA, which produces an mRNA molecule that has the same sequences as the coding strand of DNA. Although the RNA has uracil substituted for thymine, the sequence of the coding strand is usually reported when dealing with DNA sequence data, as this permits is reference to the sequence of the RNA. A site for starting transcription is required, and this encompasses a region that binds RNA polymerase known as a promoter. So promoter you can see as a P here, and a specific start point for transcription or TC. So a stop site for transcription means TC. Here, start site and stop site for transcription. From TC start to TC stop is sometimes called the transcriptional unit. That is the DNA origin that is copied into RNA and mRNA. So within this transcriptional unit, there may be regulatory sites for translation, namely a start site or TL. and a stops signal or TL. Here is a start signal and here is a stop signal. So other sequences involved in the control of gene expression may be present either upstream or downstream from the gene itself. Here is the downstream, and here is the upstream, and here is the downstream genes. So in prokaryotic cells, such as bacteria, genes are usually found grouped together in operons. The operon is a cluster of genes that are related, often coding for enzymes in metabolic pathway, and that are under the control of a single promoter regulatory regime. Perhaps the best known example of this arrangement is the lag operon. So, the structural genes of lag operon, lag Z, lag Y, and lag A, noted as Z, Y, and A, encode beta-galactosidase, galactoside premiase, and a transacetylase, respectively. The cluster is controlled by a promoter, or P, and an operator region, or O. The operator is the binding site for the repressor protein, encoded by the like L. So the repressor gene lies outside the operon itself and is controlled by its own promoter, or PI. Within the operon, there are three genes that code for proteins, termed structural genes, and an upstream control regime encompassing the promoter and a regulatory site called the operator. In this control regime, there is also a site that binds a complex of CAMP and CRP, which is important in positive regulation or transcription. Lying outside the operon itself is the repressor gene, which codes for a protein, for example, the lag repressor, that binds to the operator site and is responsible for negative control of the operon by blocking the binding of RNA polymerase. So the fact that structural genes in prokaryotes are often grouped together means that the transcribed mRNA may contain information for more than one protein. Such a molecule is known as a polycistronic mRNA, with the term cistrone equating to the gene as we have defined it, encoding one protein. Thus, much of the genetic information in bacteria is expressed via polycistronic mRNAs, whose synthesis is regulated in accordance with the needs of the cell at any given time. The system is flexible and efficient, and it enables the cell to adapt quickly to changing environmental conditions. A major defining feature of eukaryotic cells is the presence of a membrane-bound nucleus, within which the DNA is stored in the form of chromosomes. Transcription therefore occurs within the nucleus and is separated from the site of translation, which is in the cytoplasm. The picture is complicated further by the presence of genetic information in mitochondria, plant and animal cells, and chloroplasts, plant cells only, which have their own separate genomes that specify many of the components required by these organelles. So, this compartmentalization has important consequences for regulation, both genetic and metabolic, and thus gene structure and function in eukaryotes are more complex than in prokaryotes. The most startling discovery concerning eukaryotic genes was made in 1977 when it became clear that eukaryotic genes contain extra pieces of DNA that didn't appear in the mRNA that the gene encoded. These sequences are known as inverting sequences or introns, with the sequences that will make up the mRNA being called exons. In many cases, the number and total length of the introns exceed that the exons, as in the chicken ovalbumin gene, which has a total of 7 introns making up more than 75% of the gene. As our knowledge has developed, it has become clear that eukaryotic genes are often extremely complex and may be very large in depth. So, the gene size are given in kilobases pairs. The number of exons is shown, and the percentage of the gene that is represented by these exons is given in the final column. So at the other end of the scale, the dystrophin gene is spread over 2.5 megabases, yeah, 2.5 megabases of DNA on the X chromosome, with 79 exons representing only 0.6% of this length of DNA. The presence of introns obviously has important implications for the expression of genetic information in eukaryotes, in that the introns must be removed before the mRNA can be translated. This is carried out in the nucleus, where the introns are spliced out of the primary transcript. Further intranuclear modification includes the addition of a cap at the 5' terminus and a tail of adenine residues at the 3' terminus. These modifications are part of what is known as RNA processing, and the end product is a fully functional mRNA that is ready for export to the cytoplasm for translation. The structures of the mammalian beta-globin gene and its processed mRNA are outlined in Figure 2.8 to illustrate the eukaryotic gene structure and RNA processing. So, these two processes are the critical steps involved in producing functional proteins in the cell. Transcription involves synthesis of an RNA from the DNA template provided by the non-coding strand of the transcriptional unit in question. The enzyme responsible is RNA polymerase or DNA-dependent RNA polymerase. In prokaryotes, there is a single RNA polymerase enzyme, but in eukaryotes, there are three types of RNA polymerase that we know: RNA polymerase 1, 2, and 3. These synthesize ribosomal messenger and transfer 5' S ribosomal RNAs, respectively. All RNA polymerases are large multi-subunit proteins with a relative molecular mass of around 500,000. So, transcription has several component stages. 1. DNA RNA polymerase binding. 2. Chain initiation. 3. Chain elongation. And 4. Chain termination and release of the RNA. Promoter structure is important in determining the binding of RNA polymerase, but will not be dealt with here. When the RNA molecule is released, it may be immediately available for translation, as in prokaryotes, or it may be processed and exported to the cytoplasm, as in eukaryotes, before translation occurs. Translation requires an mRNA molecule, a supply of charged tRNAs, tRNA molecules with their associated amino acid residue. So, and ribosomes composed of ribosomal RNA or RNA and ribosomal proteins. The ribosomes are the sites where protein synthesis occurs. In prokaryotes, ribosomes are composed of three RNAs and some 52 different ribosomal proteins. The ribosome is a complex structure that essentially acts as a GIG that hold the mRNA in place so that the codons may be matched up with the appropriate anticodon on the tRNA, thus ensuring that the correct amino acid is inserted in the growing polypeptide chain. The mRNA molecule is translated in a 5' to 3' direction, corresponding to polypeptide elongation from N terminus to C terminus. Okay, so you can see here transcription and translation. Transcription involves synthesis of mRNA by RNA polymerase. Part of the DNA mRNA sequence is given. The mRNA has the same sequence as the coding strand in DNA or the non-template strand, apart from U being substituted for T. The ribosome is the site of translation and is made up of the large subunit or LSU, large subunit, and a small subunit, and a small subunit or SSU, each made up of ribosomal RNA molecules and many different proteins. There are three sites within the ribosomes: the A amino acid and P peptidyl sites are involved in insertion of the correct tRNA amino acid complex in the growing polypeptide chain. The finally, finally, the mRNA is being translated. The amino acid residue is inserted into the protein and in ribosome through the codon anticodon recognition event. in the ribosome. So the first amino acid residue is encoded by AUG in the mRNA or tRNA anticodon TAC, which specifies methionine. The remainder of the sequence is translated in a similar way. The ribosome translates the mRNA in a 5' to 3' direction, with the polypeptide growing from its N-terminus. So the residues in the polypeptide chain are joined together by peptide bonds. So here is the final concept map. The genetic material is usually DNA, also can be RNA. Also, the DNA gives rise to RNA. But about the DNA, arranged as a double helix composed of sugar phosphate backbone plus nitrogen bases. And so these nitrogen bases known as purines for adenine and guanine and pyrimidines for cytosine and thymine. In RNA we have uracil, so which pair up A to T, G to C, and G to U. But here is wrong because the G to C has three bonds, hydrogen bonds. Okay, but in RNA, which is of three types, rRNA or ribosomal RNA, messenger RNA or mRNA, and transfer RNA or tRNA. rRNA components of ribosomes, messenger RNA for codons, and also tRNA for anticodons and amino acids peptides, so which are involved in translation. Okay, here the genetic material is arranged as genes, gene expression, and gene structure. Gene expression requires transcription, translation, which produce protein, and also the gene structure. In prokaryotes, relatively simple genes grouped in operons, and in eukaryotes is very complex, and genes have introns, exons in 5' at 5' region cap, and at 3' we have poly A. Okay, here is the end of our class. Thank you for your attention and see you later.